All right, good morning everyone, and welcome to our session, Xenotransplant, the clinical reality emerges. My name is Riella. I'm the medical director of the kidney transplant program at Mass General, and it's a privilege to moderate this timely and transformative conversation. As you know, organ transplantation saves thousands of lives annually, but we have a major bottleneck. We have a limitation in the number of organs available for our patients. Today in the US, there's about over 100,000 patients on the waiting list, and we only perform about 40,000 transplants a year. Every day, there are about 17 patients that die on the waiting list for their promised kidney transplant. But what if we could change that completely? If we can remove this bottleneck of organ availability? And this is what we are be discussing today, xenotransplantation. So using animal organs as a source of organs for transplantation going forward. And I think we are so excited having this conversation today because if we go back maybe 5 years ago, we had not envisioned to be where we are right now in bringing xenotransplantation to the clinic. In 2024, March, beginning of the year, we performed the first xeno-human kidney transplant, or the gene-edited pig kidney into a human in the world, and the organ functioned for about 2 months. And this was just the beginning of subsequent transplants that we'll talk about going forward. And I have an incredible panel today because this is not only about the scientific development of xenotransplantation, but we're going to be talking about how can we make this a reality. So what are the regulatory, commercial, and ethical principles that we should advance in order to get there? And I'm joined by three incredible individuals here on my right. They're uniquely positioned to answer some of these questions. Mike Curtis, the CEO of eGenesis, which is at the forefront of genetic engineering of organs. Pablo Legorreta, the founder and CEO of Royalty Pharma, who has innovative ways of kind of funding research as well as advancing that to the clinic. And Julie Sunderland, founder and president of Riella Consulting, who has also shaped biotech strategies at the highest level across funding, policy, and social impact, and who also had a participation in eGenesis from the beginning. And without further ado, maybe we'll start with Mike. So you're leading one of the companies at the forefront of xenotransplantation and moving this from the lab to the clinic. And so from your vantage point, what do you see as the most effective clinical models? And tell us a little bit about your story of how do we get here?
Great. Yeah, thank you, Leo, and thanks everyone for your interest. So I've been with eGenesis now about 5 years. Came from biotech pharma about 35 years, big pharma, small biotech. And when I first heard about this idea of xenotransplantation or cross-species transplantation, my first question was just how far away are we? This has been a vision since the dawn of transplant. And I was really encouraged by the early primate data. So when you think about, how did we get here? It was a long series of primate transplants. We published those in Nature back in 2023. And that data really set the foundation for what we did, as Leo mentioned, with Mr. Slayman in the spring of 24. I think what's also really encouraging with the field is we've started to pursue this model we refer to as the decedent model, right? So we start to do transplants in patients that are in brain death. And the non-human primate data combined with these decedent subject studies gives us enough data to do a transplant in Mr. Slayman. And now what we're seeing is Mr. Slayman's results lead to the next transplant, and those results lead to the next transplant. So you're sort of getting this snowball. And so when I joined the company 5 years ago, and I think Leo's absolutely right, we never envisioned it would move as quickly as it has moved. And it's really because of the data, right? So when, unfortunately, Mr. Slayman passed away about 2 months post-transplant, but during those 2 months, the kidney functioned beautifully. I wouldn't say better than anyone expected because no one really knew what to expect. We saw 2 years of outcome in the monkey, which again led us to do the human study. So I think what's amazing of where we are today compared to 5 years ago is now we have a path from the current genetics to second generation genetics to bring those into the clinic. Many people in the field felt like we would never be in the clinic. Like this would never, I think there was an old adage that xenotransplantation is the future and it always will be. Like that's no longer the case. Like it is here and we're actually helping patients.
Wonderful. Julie, I'll get your perspective now because you were in the beginning of eGenesis. So provide us a little background and how you see things going from there.
Sure. So I'm remembering back almost 10 years when we started eGenesis, and it literally was Luhan Young, who was a postdoc in George Church's lab, and I remember going out to Longwood and visiting her in the Weiss Institute, and when we actually did the transplant with Mr. Slayman. It was such an extraordinary time. It was such an extraordinary milestone for the company. And I was on the phone with George Church, who is a character for those of you who know him. And we were remarking on how at the beginning we had to have kind of a delusional belief that this was going to work, that gene editing and that multiplexing editing that George and Luhan had worked on was going to allow us to do these extraordinary edits that would make the organ as compatible with humans as possible. And we went through so many challenges. I was telling Leo and Mike and Pablo about the first time we actually got a live pig and how that was a huge milestone. We had a board meeting a week later, and unfortunately the pig passed away, and we were all devastated, and we thought we were going to have a birthday party, and we ended up having a funeral for the pig. But to go forward a decade and to see us go from solving problem after problem, getting the genetic construct right, getting the production of the pigs right, getting those non-human primate trials, working so closely with our extraordinary colleagues, both at NGH and at other academic centers, problem after problem after problem. And George and I were reflecting that we had to actually believe 10 years ago that it was going to work, but we had a small little voice in the back of our head that, oh, this is never going to work. And so to have that first and that second and that third transplant and have these patients do so well, we were just like, oh my gosh, I cannot believe this is actually working. And so it's just such an exciting moment. It has taken so much work from so many people to get us to this point, and I do believe that we are at the turning point where we will have ubiquitous organs that will save countless patients' lives and make their lives better too.
Thank you, Julie. Pablo, give us a little bit of a background, and also if you could share your personal story of why this is such an important topic for you of investment and research.
Sure. Good morning, and of course, Leo, and thank you very much. Leo and I have a personal connection because he is the transplant nephrologist of my son who had a transplant at Mass General Hospital in May of 2022. It is a very personal story for me because Pablo, my son, who's now 27, was born with urinary reflux, and we knew from birth he had a surgery to correct reflux at Boston Children's, but we knew that eventually he was going to need a kidney because his kidneys were damaged because of the reflux and infections. And thanks to what I do, running Royalty Pharma, which I've been for 28 years, and this company now invests in royalties, and for the last 12 years we have been funding late-stage trials for biotech and pharma at scale, $27 billion invested, and we end up with a royalty. But obviously, my exposure to the life sciences research ecosystem gave me this ability to actually understand what was happening in transplantation and to potentially benefit my son. So my first involvement with the Mass General team was because I scoured the planet to see what was happening in tolerance to improve outcomes after transplantation, essentially get rid of immunosuppressants. And what I found was that the Mass General team had by far the best approach. Stanford was working on it, Northwestern and others, but MGH had the best approach. And I got involved with them in 2018, early 18, with an antibody, anti-CD2, that is critical to induce tolerance, to condition a patient 7 days before the transplant, to prepare the patient so that the day of the transplant, like my son, who got two amazing things from my wife, her kidney, and also bone marrow, and this bone marrow that came in, which brought in my wife's immune system, educated Pablo's immune system to accept her kidney and not reject it. So Pablo became tolerant, and he is tolerant now, and he was weaned off from immunosuppressants. He was put back on a tiny amount because a COVID vaccine revved up his immune system, and somehow, there was some concern. It turned out that it was the COVID vaccine, nothing else. So hopefully he will be weaned off eventually. But that's how I got involved, and obviously for my son, this has been an incredible outcome because he has a chance now of a normal life, no immunosuppressants, and a kidney that could work a lot longer than others because of no immunosuppressants. But that was my initial involvement, and David Sachs, who is the individual with the team at Mass General, Tatsuo Kawai also, who had developed over 40 years this approach to induce tolerance and get rid of immunosuppressants, had another project, which was he worked for 40 years on a colony of pigs that he inbred over 40 years, very patiently, very meticulously, to knock out the genes that cause rejection in humans, alpha-gal. What eGenesis is doing, and Mike and Julia are involved with, is they're doing it through gene editing. David do it, in the more classic way by inbreeding the pigs. So our colony doesn't express, the pigs, don't express this gene, and they're very special. And then other things that David has done is that he invented this thing called the thymokidney, where he takes the pig's thymus and actually, takes a piece of it, minces it up, and then through a surgery, they put the thymus 1 month before the kidney is going to be used, the pig kidney. The thymus is put under the kidney capsule, and the pig is closed up again, and over a month, this thymus under the kidney capsule, it's magic what happens. The thymus sort of revascularizes, reorganizes itself, and now we have this construct that actually is potentially capable of also inducing tolerance, again, to minimize immunosuppression. So we've come a long, long way. And because I was David Sachs' partner, and I saw what he had done, and there was a point in time in 2019, 2020, where the funding was drying up, and I just looked at his incredible feat, and I said, this cannot be lost for humanity. 40 years of work, this incredible colony. So I decided to get involved on that project also with him personally. And what's great, Mike alluded to this, is that after 40 years of work, in March of this year, we took one of our pig kidneys and put it into a decedent, brain-dead human at NYU Langone. It worked beautifully. There was no sign of rejection. Obviously, the kidney was taken out after some days, 22 days, because the family wants the body back to bury it eventually. But we've also filed an IND with FDA to start a phase 1 trial in humans with our pig kidneys. And it's just so exciting to me because, as you said, we're really at the cusp of potentially delivering organs, kidneys, livers, and other organs to millions of patients, really, when you think of the world that actually will benefit from this. And I think one of the things that's interesting about David's colony is that because they're inbred, and we have organs that potentially are going to be at a very low cost because we don't need to do the gene editing. So, it's exciting, and happy to engage later in other topics with the panel and all of you.
Thank you, Pablo. I think Pablo touched on the two major challenges that we have in transplantation. We spoke about the organ availability, but the second one is, the anti-rejection meds and how can we minimize that in the long term. So if we think about that, Mike, I'm going to turn it to you because your pig now is the most gene-edited pig across all the companies currently working on, and the goal of these gene edits goes from reducing the infection risk and then increasing compatibility. We have a product at hand moving to the clinic. So how do you envision then we improving upon this, and how would this be done in humans? Would this need to go back to the non-human primates?
Yeah, no, it's a great question, and something we've thought about since the beginning of eGenesis. So, we've leveraged discoveries over the past several decades into this single animal that is currently in the clinic. And so when we think about genetic engineering, it's really three different classes of edits. It's the inactivation of genes that lead to hyperacute rejection. We typically refer to this triple knockout. Then we introduce human regulatory genes. In the case of our clinical donor, it's 7 of those genes to control complement activation, coagulation, and innate adaptive immunity. And then the third class is the inactivation of endogenous retroviruses that are present in pigs. If you rewind the clock to the mid-90s, was discovered that porcine retroviruses can infect humans, it really put a chill on the field because no one wants to be part of a zoonotic spillover from pigs into humans. And so because those retroviruses are integrated into the porcine genome, 50 to 70 copies, there was really no technology that was available that could really produce what we call retrovirally inactivated animals or completely safe pigs. Along comes CRISPR-Cas9. It's the ideal tool. This is what Lujan and George initially showed, is that they could use it to inactivate viruses. And then we optimized in the monkey. And because we have this amazing genetic engineering tool, there's really no end to the amount of editing we can do. The problem is, if we optimize for the monkey, we're optimizing for the monkey. And so we maybe can produce organs for monkeys. We want to produce organs for people. And this is why it was so important to be able to push into decedent studies, into the first patients, because that feedback from the patients, we can already see it in the first few recipients. We already have a list of genetic engineering that we would like to do in the second generation animal. And because we now have this path to clinical studies, even small studies, 3, 4 patients, there's now an iteration loop that didn't exist even a year ago, that we can envision, learning from the patients, going back to the animals, going back to the immunosuppression, and optimizing. So I think we're in an optimization circuit now that, again, was only dreamed of at the beginning of this, even a year ago. And so because we can edit so quickly, I can see that the next generation will be right on the heels of first gen. But what we've already seen in the first gen, kidneys, I believe are going to help patients with this first gen product, but our ultimate vision for engineering the pig is to produce organs that don't require suppression. And we believe the way to do that is through iterative engineering.
And I think one of the unique pathways of Xeno that's very different than drug development is that it was started as a compassionate use application. So you're kind of getting the sickest of the sick patient, attempting to perform the transplant based on all the challenges that were existed. But quickly, the FDA and their members allow us then to move on into these small pilot studies that allow us to reduce the risk of these patients. So starting with patients that are not as sick, and this is really important whenever you're testing a drug or a treatment like this, because you want to avoid all the confounding factors that may come if you're getting too sick of a patient, starting from the beginning. Julie, I think one of the key questions that I think starting to become centerfront from everything that we do is, what defines success in xenotransplantation?
Yeah, I think that Mike's already touched on it a bit, and Pablo's already touched on it a bit, which is ultimately we want to get to the point where we can have ubiquitous organs that are accessible at an appropriate price for as many patients as possible without immunosuppression. I do think that that's a pathway. So if we look at what we're doing today, it is a comparable organ. It's got some differences that I think the clinical team is working out, but that's where we are today. If you look at the power of editing and the ability to really understand what's going on in the pig genome and figure out how to make those organs more and more human compatible, we have a lot that we can do. I would say that the biggest challenge right now is that regulatory framework. It's been great to have the openness of the agency to the compassionate use as well as moving towards INDs, but our ability to iterate on that, on the product to get it more and more compatible for humans, I think is the ultimate goal here. The other thing that I would say as success really comes back, I would almost throw that back to you, Leo, and say, one of the things that's been incredibly encouraging over the last couple years is the degree to which the patient community has really been active in wanting to support this process, to support the company. If I look at our first 3 patients, those guys are heroes in terms of their willingness to come forward and do something so experimental. What has been really interesting, I think an eye-opener both for us as well as for the regulators, is the degree to which this is not, they don't view this as an option that is sort of the last resort. They view this as an option around quality of life. And if you look at their life on dialysis, we've had patients basically say, we don't want to be on dialysis. We would rather take the risk of a xenotransplant than experience the loss of quality of life on dialysis. And so for us, I think that really is a North Star, in that we are looking at how can we really improve quality of life here relative to dialysis, which gets back to that long-term pushing for both a robust organs, but also reducing immunosuppression to get to that quality of life. But Leo, I'd love to hear your view on patients.
We were also so surprised by the number of messages, by the number of people reaching out to us after we did the first Xeno and offering to be volunteers. And if you can imagine, we had a consent form of over 40 pages. And the toughest question that we had was, will this kidney last? And the answer is, we don't know. We had all these non-human primate data, monkey data, that some organs would go above 2 years. But we could not say for any of these patients that the kidney was going to work for a long time and that they were going to be better off than dialysis. But they were willing to take the risk. And this was not 1 or 2 patients. We got hundreds of messages of people volunteering to be part and receive the xenotransplant. So what it tells us, on the physician side as well, is that our patients are very unhappy with their treatment, which is dialysis. So dialysis was meant to be a short-term treatment until the patient gets transplanted. But now we have in the US more than 600,000 patients on dialysis, and for most of these patients, it's their permanent, their lifelong treatment. So suddenly, what we thought as a bridge, which was a dialysis, now it becomes this permanent, and the quality of life and how the wear and tear happens with dialysis and the overall health is major. And the patient advocacy, so patients actually speaking out and saying, look, we want alternatives. We want the field to advance and provide us with something other than dialysis. And this has allowed also the regulatory agencies to rethink about how can they help us advance and move this forward. But without the patients, without true heroes and pioneers that have actually volunteered to be part of this, this would not be possible. Pablo, we'll talk about success. What is in your perspective?
So maybe just to add a little bit to the alternative dialysis. In the US, you mentioned the 600,000 patients that are on dialysis currently. There's about 135,000 patients that come into dialysis every year that have kidney failure. Only 5,000 of the 135,000 will get a kidney, and 130 don't get a kidney and then move into dialysis with an average survival of 5 years. So it's not really a lifelong solution, because many of these patients don't do well on dialysis. Many even give up dialysis. Stop dialysis, knowing that it's going to be dire, but they just don't want to continue. So it is a terrible alternative. Now, it is the #1 category of expenditure of the US government, more than $150 billion per year spent on dialysis. Private insurers spend even more. But every government around the world, #1 category of expenditure is dialysis. So what we're trying to come up here with is a solution to this very bad alternative. And in terms of success, when I think about it, I think this will evolve like transplantation did decades ago, where the first transplant lasted a small amount of time, and then the envelope was pushed and pushed and pushed. And now, in terms of allotransplants, human to human in kidney, for example, you have statistics that point to amazing success. 2/3 of the kidneys survive 10 years or more, and the median survival is 12, 14 years, and depends whether it's living or deceased donor. Living donors, much longer. If you're healthier and younger, much longer. But I think for this to really succeed and get the support of the ecosystem, payers and insurers, and we probably are going to need to have organs that last a couple of years, 6 months. The FDA has been very flexible, as Leo and others pointed, because they have given us a primary endpoint of 6-month survival, which is actually great, but it's the beginning, right? And I think for this to really be acceptable by patients, by the hospitals, by the doctors, by payers, we probably need to have organs that are going to last a couple of years, 2-3 years, and then we will, through a lot of additional work, we will start to push the envelope more and see if we can get them to last even longer than that. And I honestly, looking at the data we have in baboons, I think it's likely that these organs will last as long as a year or two initially, maybe more. And I think one of the critical things will be figuring out how to make these patients tolerant, like what happened with my son, with an approach that actually combined, the transplant with bone marrow and, also a conditioning of his thymus, which is critical. And that's why David came up with this idea of creating this thymokidney. And that, I think, might get us to a situation where we can really start to lower immunosuppressants and have these organs last for a long time.
So Mike, linking the success to bringing this to commercialization. So what are the challenges there? Who's going to pay for this, and what other roadblocks you may see on the way?
And I think Pablo's already kind of hit on one of the big ones. What's the outcome, right? And I think we were really pleased that the FDA agreed to a 6-month primary. But to Pablo's point, for patients, for payers, we're going to need to see longer. And in our trial, it's a 6-month mean graft function. We expect patients to go longer, and Mr. Andrews, our first patient, is already at 8 months. So we've already surpassed the endpoint, and the goal really is to go much longer. What we're also thrilled by was in a relatively small number of patients, and our trial is 33 patients, we can, I think, demonstrate efficacy and help us create a label. And I think it's, again, with 33 patients, it's clearly not for all 600,000 patients on dialysis, but for patients who meet the eligibility criteria, right, older patients, patients who are progressing on dialysis and not doing well, I think that's the commercial play early on. I think the challenge, we can currently make a few hundred animals per year. We're currently planning to make a few thousand, so there's definitely a supply challenge. But the good news is, in the United States, we make about 130 million pigs a year. So the agriculture industry has figured out how to make animals at scale. So the scaling issue, I think, unlike things like CAR T and cell therapies, which has been a problem, I don't think we're going to have the same kind of issues because the road has been paved of how to make pigs at scale. So I think that's sort of solved, or relatively, nothing's easy, relatively straightforward. The next piece is the payer piece. What are people willing to pay? And I think you can look at the economics of dialysis, you can look at the outcomes in dialysis, and you can get yourself into the range of several $100,000 per kidney, depending on how long they last. If I can give a patient a 5-year kidney, there's a price. If it's a 2-year kidney, there's a different price. And so, but we were already having the discussions with payers, both government payers and private payers, and to Pablo's point, their mission is not only because it will help patients to get them off dialysis, but will also, help the budget. And so, dialysis is, suboptimal care, but it's the only thing these patients have. Getting them off dialysis, getting them into a transplant is the goal. And I think everyone, regulators, payers, and us as the innovators and patients are all aligned on that same goal. So I think we are now on a path, and we're actually laying this out, BLA and, a path to approval by 2029. Which, again, rewind the clock 5 years, no one ever thought we'd even be thinking about a registration trial, and here we are starting one.
Incredible. Julie, could you share with us your thoughts about scalability of this?
Yeah, I think Mike has presented a very optimistic case, which is...
That's my job.
Again, if I go back to the history of this company, when we were starting to try to figure out how to, produce pigs, I actually grew up on a farm in Nova Scotia with pigs, and I swore I was going to get out of there and never go back. And, 30 years later, I find myself as a pig farmer. But that challenge of producing animals at a quality level that is acceptable to regulators, that is definitely a challenge. There's a scalability issue to this, and I think we have to figure it out. And we have, a group of people around the table that can help figure that out. It's been done before, so it's not an impossible challenge. It just is more complicated. If I think forward 10 years, I can imagine, multiple production sources managed by some of these experts in animal husbandry, linked to transplant centers, delivering organs on an as-needed basis. And that seems like a big vision and one that's going to be hard to execute on. But if you actually look at the reality of transplants today and how complicated and difficult it is to get organs to patients, it's certainly surmountable and something that we're prepared to do.
I think, Leo, talking about scaling, what's interesting is that the pig, the swine, is really the ideal species because, and particularly the ones that both eGenesis and Corianex, the company I'm involved with, we're working with, are the miniature swine that get to be about 200 pounds, 100 kilos, fully grown. The normal pig, the wild pig, is more like 1,000 pounds, too big, and the organs are too big for humans. But David Sachs and also eGenesis had this idea of working with miniature swines where the organ is the same size as a human. You can't tell the difference between a pig kidney and a human kidney if you have two of them side by side. But it's the ideal species because the gestation period is about 3 months, and a little bit more than 3 months. So you could have a sow give pigs, twice a year, maybe 3 times a year. And every time a litter would produce, 6 pigs, 8 pigs, depending on certain factors. So every time, you will have 6 pigs with 2 organs. It's tricky because to plan 2 transplants at the same time, if a pig has 2 kidneys, is very tricky, but at least 1 kidney from 1 pig initially. So it is the ideal species really to scale this up. And I could foresee a future, I don't know if in 10 years, maybe a little bit longer than that, where you could have tens of thousands or even hundreds of thousands of animals producing organs for the world. Anyway, that's the vision. And I think what's so exciting is that it's happening today. I mean, we're in humans now. So at this point, it's all about, making sure we do the right thing, we don't make mistakes, that we optimize, the animals, and then, over time, let's get them approved by FDA. One of the things that was mentioned that happened also, thanks to data that has been presented, is that the FDA has lowered the bar for us and not forced us to really work with the sickest patients. And essentially what the FDA has realized that if you're a person on dialysis, and if you actually can demonstrate that your chances of getting an organ in 5 years is less than 10%, you would qualify. So you could be healthy, just on dialysis, but not have, like Slayman, the first patient, was a patient with a lot of complications, cardiovascular issues and others. Now we're starting to work with patients that are actually in much better shape, which will allow us to really judge how well these organs are working. So there's a lot of things that are happening that are really positive for the field.
Amazing. We have only a few minutes left. There's so much to talk about. I'll tell you that from the clinical perspective, looking at the blood work of our patients on a weekly basis and seeing that the pig kidney is able to perform exactly like a human kidney in terms of the balance of all the minerals, the water balance, hormone production, blood pressure control. It's just out of this world. So we're so fortunate, I think, to be living this. But of course, if you ask me, what do I envision 10, 15 years from now, Mike? I think one of the things that we would love to do is use the gene edits to personalize an organ so that this organ is going to be produced for this specific patient, so that we can overcome whatever immunological barriers exist and create this invisible organ, an organ where the immune system of the recipient will not be able to identify it as a threat. So would that be possible?
Yeah, no, one thing I've definitely learned about being in this field is never say never, because it will happen. And I think, working with folks like George and innovators in genome engineering, in the iteration of the ability to see a patient, and if we have time, right, we can definitely do that. And I think it's just a matter of knowing what edits to make, and I think this early clinical data will help us do that, and continued advancements in genome engineering. And so what are the bits of the porcine genome that we need to make more human-like to get to that point, to make, tailored custom organs? When we first entered this field, people felt like you couldn't, for instance, make animals by cloning at any kind of scale. That's all that we do at eGenesis. So we make hundreds of animals each year by cloning. And now we think we have a visibility into making thousands or tens of thousands of animals by cloning. 5 years ago, people said that was impossible. And not only is it possible, we were doing it. Right? So I think, tailored, customized organs are absolutely the future. And how we get there, we don't quite know yet. But again, we never say never.
Amazing. So we're getting to the end of the hour. Is there any burning question from our audience?
Yeah. So we believe we just started it. So it's a phaseless, we're calling phaseless, so phase 1, 2, 3, 30 patients, powered at 50% of patients hitting a mean graft function at 6 months. We think that's it. Now, in that data set, we expect to have patients longer than 6 months, but if monkeys are any indicator, we may also have patients that are shorter than 6 months, but the mean at 6 months. So that's the ingoing, and they didn't say no.
Incredible.
Mike, they said yes, just to be really clear.
They did say yes. The detail, the beauty will be in the data, how things go, but yeah. So we feel like we have a path to registration.
Awesome. So this was a great conversation. The hour, it passed so quickly that we're already on the top of the hour, but it was great to get the insight, I think, from leaders in the field and really talking about something that, again, was unimaginable 10 years ago, and now we're talking about xenotransplantation as a clinical reality. Thank you all for attending, and I hope you enjoyed the rest of the conference.